Good morning everyone. Today I will be discussing about this different staining techniques in microbiology. Firstly, let's have an overview. Firstly, we'll discuss about the introduction, then fixation, then after that dyes used in staining, and last one staining techniques. So firstly, introduction, it will include the microbiology definition, in which the major question that comes is why do we need to stain bacteria? Now firstly, what is microbiology? Microbiology is a field of biology that deals with the study of microorganisms. It basically involves the study of agents or organisms that are too small to be seen by an unaided eye. So they are required to be seen in the microscope. Now the next questions come, why do we need stain bacteria? Why do we need to stain bacteria? So as all of us know that bacteria, they have same refractive index as that of water. So they when we observe in the microscope, usually in the under light microscope, they appear opaque. They appear opaque. That is, they are literally invisible when we observe them without staining. So staining is an essential part of staining is an essential part to observe the microscope, to observe microorganisms under microscope. Now although the microorganisms, they can be easily seen under light microscope, but they need to be fixed. Firstly, they need to be fixed in a certain position so that we can observe them easily under light microscope, but we need to fix and stain so to observe under the light microscope. Now next is the fixation. Now what is fixation? Fixation is basically a process of fixing cells in a certain position, in a fixed position, and preserve the cell structures in a certain position so that they can be fixed in a position while staining. Now there are basically two types of fixation. First one is heat fixation, and the second one is chemical fixation. Now what is heat fixation? In heat fixation, firstly a smear is prepared, a bacterial smear is prepared. For example, smear is generally can be prepared from two types of media. First one is your liquid media and second one is your solid culture media. So when we prepare a smear from liquid media, we don't need to add distilled water because it's already in a liquid culture. And secondly, if we want to make a smear from solid culture media, we need to add distilled water before making this smear or before preparing this smear, because it is in a solid culture and cannot be observed in the microscope. So in heat fixation, generally firstly smear is prepared, which is a bacterial or any other microorganism smear. So after the preparing smear, after preparation of the smear, it is heat fixed. Firstly it is heat fixed, that is, the smear, the slide containing smear is passed over the Bunsen burner frame. It is gently passed over the flame of a Bunsen burner. Generally it is used to observe prokaryotic cells, and it is generally used to observe prokaryotic cells. And secondly, the main feature of this fixation is that it only preserves the cell morphology, but not the internal structures, that is the structures which are present within the cells. Now, the second type of fixation is the chemical fixation. In this, certain different chemicals are used for fixing the smear. Firstly, for example, carbon, for example, ethanol. The example of different chemicals which are used for chemical fixation of the bacteria are ethanol, acetic acid and formaldehyde. So, all these three agents are used for chemical fixation. The major difference between the chemical fixation and heat fixation is that unlike heat fixation, which only preserves the cell morphology, so in chemical fixation it is both the cell morphology as well as the internal structures, the structures present within the cells, they are preserved. So, this method, chemical fixation, it is used for fixing which are microorganisms that are more delicate. Now, next is your dyes. Now, dyes they have certain chromophore groups which bind with the cells by ionic bond or through covalent bonding. So, in dyes they have basically chromophore groups, and they bind with the cell and the microorganism through ionic bonding and covalent bonding. Generally, these chromophore groups which are present in these dyes, they provide a color to the dye, and in dyes there are basically two types of dyes that can be used. They are, first one is basic dye and second one is the acetic dye. So, let us come to the basic dye. The basic dyes, they are generally positively charged dyes. They have positively charged group that bind to the negatively charged cell components, like they bind to the negatively charged cell components like nucleic acids and proteins. Now, some of the basic dyes that are used are example of basic dye like methylene blue, which is a basic dye. Now, next one is the safranine. So, methylene blue, safranine, then malachite green. The most major dye which is used, basic dye which is used in staining techniques is the crystal violet dye. And last one is the basic fustian. So, these 5 dyes are most commonly used as a basic dye for staining techniques. Now, the next dye is the acidic dye. So, these dyes, they are negatively charged. They are negatively charged dyes and they contain negatively charged groups like carboxyl groups and hydroxyl groups. So, these negatively charged groups, that is the carboxyl group and the hydroxyl group, what they do is they bind with the positively charged cell components, for example capsules. So, they are positively charged and they bind to the they are negatively charged and they bind to the positively charged cell components like capsules. Now, examples of acidic dyes, the most commonly used acidic dyes are eosine and acidic fustian. Now, next one, the last content of this discussion is the staining techniques. Mainly there are three types of staining techniques. First one is the simple staining method, and the second one is the differential staining, and the last one is the special staining. So let us firstly discuss the simple staining. So in simple staining only single dye is used, and this simple staining technique, it is generally used for observing the prokaryotes, that is the prokaryotic cells. So simple staining is generally used for prokaryotes, and the method that is followed in simple staining is that firstly a bacterial smear is prepared, which is then air dried. Firstly the smear is air dried, and then after that it is heat fixed, that is by it is passed over the flame, and then after that it is kept still for 1 minute for the cooling down of the smear, and then after that it is flooded, the smear is flooded with the any of the basic dye, that is carbon fustian, or we can use the basic fustian or crystal violet. So after that, after flooding with the basic dye, it is washed under tap water. We do not need to wash it with the distilled water, it is simply washed under running tap water, but we have to make sure that the pressure of the water is not too high, so that the smear is not washed away with the tap water. Now simple technique, in simple staining technique, it is used for observing the cell morphology and not the internal structures of the cells. And one of the example of simple staining is the Bacillus anthracis bacteria, which is it is stained by methylene blue stain, that is your basic stain, basic dye. So it is stained by methylene blue. And another example is of Vibrio cholera. So Vibrio cholera is stained with carbon fustian. Now next one is after the simple staining, let us come to the differential staining. Now what is the differential staining? Basically differential staining techniques are those techniques that divide the different type of cells or bacteria into different groups. That is they categorize the cells into different groups or types, and there are the two types of differential staining are, first one is the gram staining. And second one is the acid fast staining. So both these staining techniques, that is the simple and the differential, they preserve the cell morphology, that is the shape, size and arrangement of the cells as well as the internal structures of the cells. So firstly in gram staining, this technique was developed by Hans Christian Gram in the year 1884. So this technique is the most commonly employed technique in bacteriology, and it was developed by Hans Christian Gram in the year 1884, and it is basically used to study the morphology of the cells, and secondly it is used for differentiating the bacterial cells into two types of categories. That first one is your gram positive bacteria and second one is the gram negative bacteria. So it differentiates the bacterial cells into basically two types, gram positive and gram negative. Gram positive bacteria and gram negative bacteria. The procedure which we follow in gram staining is firstly that a smear is prepared, same bacterial smear is prepared. It is in second step the smear is it is air dried. Firstly the smear is air dried and then secondly smear is heat fixed, so that all the cells in the smear they are fixed properly into the slide so that we can observe them easily under the microscope. So in the first step of gram staining procedure is that firstly the smear is flooded with crystal violet dye, that is the primary stain. The first step is a smear is prepared, it is firstly treated with crystal violet dye, that is it is a primary stain. Crystal violet is a primary stain. Now crystal violet, it also acts as a simple stain, as it can be used in staining all the types of cells. So after flooding the smear with crystal violet, it is kept still for 1 minute, after which it is washed under the running tap water. So it acts as a primary stain, crystal violet. Now after treating the smear with crystal violet, it is in the second step, the smear is treated with the grams iodine, which acts as a mordant. The grams iodine is a mordant. And now what is mordant? Mordant is basically the agent which is used to increase the cell affinity for stain. That is, it binds with the primary stain, that is the crystal violet stain, and it forms a complex that is trapped in the cell wall of the bacteria. So secondly, mordant is used, gram iodine, that is. We follow the same procedure as the in case of crystal violet. We flood the smear with grams iodine and keep it still for 1 minute and then wash it under the running tap water. Now the third step includes the decolorization. The third step is the decolorization step. What this step does is that this decolorization, different decolorizers are used, for example alcohol or ethanol. So it removes the primary stain from the cells. So in this step the cells become colorless at this stage. This step is the major step of the gram staining, and it forms the major basis for differentiation of gram negative and gram positive bacteria. In this, the main thing to keep in mind is that we do not need to keep the slide still for 1 minute. We just have to add a drop of alcohol and then wash it the moment we put the drop. It is not kept for 1 minute. The last step is the flooding the smear with safranine, that is the counter stain. So it acts as the counter stain. So in the first step the cells remain, that is the gram negative cells and gram positive. So in the first step these cells remain purple in color. At this stage the cells are purple in color. In this stage also both the gram negative and gram positive bacteria, they are purple in color, they remain purple in color. Now in this step which forms the basis of differentiation, so in this in this step all the cells they become colorless. That is the peptidoglycan that is present in the cell wall of the gram positive bacteria, it is dehydrated by alcohol. The peptidoglycan content which is present in the cell wall of gram positive bacteria, it gets dehydrated. In this stage, that is in the last one, safranine stage, the gram negative bacteria they are counter stained with the safranine. The gram negative, they are stained with the safranine and they become pink in color, they appear pinkish or reddish in color. The color may vary, and the gram positive cells, they retain the crystal violet color. So the main difference between the gram positive and the gram negative bacteria is that of the peptidoglycan. That is, in gram positive bacteria the peptidoglycan content is higher as compared to the gram negative. In gram positive bacteria generally the 50 to 90% of the cell wall is covered with the peptidoglycan, and in the case of gram negative only 10% of the cell wall contains the peptidoglycan, and moreover the gram positive bacteria, they have very less lipid content. They lack the lipopolysaccharide layer, which is an additional feature of gram negative bacteria, and in gram negative bacteria the lipid content is quite high as compared to the gram positive. So when the crystal violet and iodine complex is formed after treating with the mordant, so what happened in case of gram positive bacteria, they since they have very less amount of lipids, phospholipids and lipopolysaccharide, and the lipopolysaccharide layer is also absent. So what happened is the crystal violet and iodine complex, it gets easily destructed, it is extracted out from the cell wall, and hence the gram positive bacteria, they retain the crystal violet color and they appear purple in color. And in case of gram negative, which has a very less, less peptide to glycan, nearly 10%, but they have very large lipid content, that is they contain a higher amount of lipids, phospholipids and lipopolysaccharide. They have an additional feature of including a lipopolysaccharide layer, the outer membrane. They have outer membrane which is separated from the inner membrane by a periplasmic space. So in case of gram negative, the crystal violet and iodine complex, it is not, it is generally disrupted, yes it is disrupted, and the cells become colorless and they are easily counterstained with the safranine. Now gram status, it is very important in medicinal purposes, because the absence and presence of the cell wall, it plays a very important role in the susceptibility of antibiotics, because the absence of or presence of cell wall, it is directly related with the susceptibility of antibiotics as far as the cells are concerned. So now the example of gram negative bacteria. Firstly, in case of bacilli, that is E. coli, they appear pink in color and they appear rod, they are rod shaped. And second example is the streptococci species. They are cocci in chains, and examples of gram positive bacteria are Staphylococcus aureus and the Clostridium and Corynebacterium. So the Staphylococcus, the species Staphylococcus aureus, they are usually cocci which are present in clusters, they are not present in chain, they are present in clusters. And the second example is that is of bacilli. So the bacilli involves Clostridium species and Corynebacterium species. Now the next type of staining, that is the differential staining, is the acid fast staining. So this method was developed by Ehrlich and it was modified by Nelson. So this method is usually used for identify two types of species of bacteria, that is Mycobacterium tuberculosis and Mycobacterium leprae. These are the two pathogens which are responsible in causing the tuberculosis disease and leprosy respectively. And Mycobacterium leprae, which causes leprosy and it causes tuberculosis. So these two strains are the non-acid fast bacteria. Now both of these bacteria, they have very high lipid content and more in particularly the mycolic acid, and so what they do is they prevent the cells from dying, that is they are not easily stained with the dyes. And now the method which is used for this identification is Zell-Nelson method. So Zell-Nelson method is used for, it is used as a acid fast staining method, and in this method there are different steps involved. For example, firstly the bacterial smear is prepared. Now in the next step, that is your second step, the smear is treated with carbylfuscin, and then after that it is heated for, in the next step, that is the third step, it is heated for 5 minutes, and then in the next step it is washed with the distilled water, and then after washing with the distilled water, it is decolorized by using 20% sulfuric acid. Then again it is washed in the fifth step, again it is washed and it is treated with the acidic alcohol. That is the sixth step, acidic alcohol. And the last step involves the counter staining, that is the cells, they are counter stained with methylene blue stain. So usually the result involves the acid fast bacteria like Streptococci species and Staphylococci species, which are acid fast bacteria. So they appear pink in color, and usually the non acid fast bacteria, that is Mycobacterium tuberculosis and Microbacterium leprae, they are blue in color, they appear blue in color, and in this method usually fluorescence microscopy is used. Now let us come to the last topic, that is the special staining. Now special staining is the staining method that are used for staining the special structures like capsules, flagella and endospores. So the staining techniques involved are capsule staining, and second one is the endospore staining, and the last one is the flagella staining, which is used for staining flagella. Thank you.